Well, welcome to the Harold Bealey Library's House of Learning lecture series. I'm Mike Hunter, the coordinator for this year's lecture series. In the Doctrine and Covenants, section 88, verse 119, it says, "Organize yourselves and establish a house, even a house of faith, a house of learning." Through this lecture series, the Harold Bealey Library attempts to establish a house of learning by bringing together scholars and students in a discussion of ideas. We're privileged to have as our featured speaker today Professor Heidi Vollmer-Snarr, who is from the BYU's Chemistry and Biochemistry Department. Professor Vollmer-Snarr moved to Utah in 1994 to speed skate, and while in Utah, she also completed her undergraduate degrees in German and chemistry at the University of Utah, where she was first introduced to organic chemistry. Professor Vollmer-Snarr began synthesizing anti-cancer compounds as an undergraduate student and decided to continue this endeavor in graduate school at Oxford University. She then did postdoctoral studies at the Sloan Kettering Cancer Center in New York and completed her postdoctoral work at Columbia University. While at Columbia, she began her work in age-related macular degeneration and met her future husband, Trent. Since coming to BYU, Trent and Heidi were married and now have two highly energetic boys, Zach, 4.5, and Grady, 2.5. Professor Vollmer-Snarr continues to do research in both cancer and vision here at BYU, and she will speak today about this research in her lecture entitled A Search for a Cure. So please welcome Professor Heidi Vollmer-Snarr.
Thank you very much for the introduction. And my punchline was going to be, so, search for a cure. You came here to hear about a cure, but a cure for what? But I guess the introduction gave that away. What I'm doing in the lab is I'm synthesizing a library of vitamin A derivatives. And hopefully by the end of this talk, you'll understand a little bit more what I mean by that and what I'm doing with these vitamin A derivatives. So, we're using these compounds in order to detect and isolate hopefully more of these compounds specifically from the human eye in an attempt to look for compounds that may be involved with macular degeneration. And we're also using these compounds in the development of a targeted and triggered drug delivery system for cancer therapy. So I'm going to start by telling you a little bit about age-related macular degeneration and then work towards, specifically telling you about the research that involves these vitamin A derivatives. So first of all, what is age-related macular degeneration? It is a disease that can lead to blindness and is actually the leading cause of blindness in developed countries. So, as you see here, the macula is the central part of the retina, which is in the back of your eye. And the disease involves the deterioration of cells in the macula, which end up resulting in a loss of the central high acuity vision. So I'll show you in a moment some photos and then show you what the photos might look like if you had AMD. So the reason why this disease is so difficult to treat is because the cells in the macula that die are postmitotic cells. And what that means is once they're non-replicating cells. So there are some cells in our body that will continue to replicate throughout our lifetime. But these cells, once they're gone, they're gone. And the cause of the disease is unknown, although I'm going to talk to you about some hypotheses that we and other labs have come up with relating to this disease. And because the cause is unknown, there's really no known cure. And by cure, I mean something that will literally bring back these cells that are no longer replicating. Okay, so here are the photos that I promised you. And I always love putting these photos up because these are my two little boys back when they were 1 and 3. And if you were suffering from age-related macular degeneration, you would not be able to clearly see, really, if you were looking at them in the face, you wouldn't be able to see their faces clearly. And you'd see something more like this, and that would obviously be very sad because they're quite cute. So, and if you were reading, the same would be true. You have some peripheral vision left, but you're not really able to use the central vision when you're afflicted by this disease. Okay, so what are the risk factors? Are you at risk for AMD, or age-related macular degeneration? As the name suggests, age is a risk factor. In fact, about 20% of the population over 60 have some form of this disease. So it may not be as severe as, those photos that I showed you where you can't make out someone's face at all. Initially, in fact, ophthalmologists will detect this disease because and I'll show you some photos of this, but they can take photos of the retina and they begin to see this cellular deterioration even before you may detect the loss in your central vision. So age is a risk factor. There are people who have macular degeneration in their youth, but those are more rare forms of the disease. Genetics also plays a role. So if you know of someone in your family who has the disease, it's likely that you could develop it, or your likelihood is much greater than maybe somebody who doesn't have anyone in their family who has the disease. Smoking habits, actually, there's a slight increase in risk if you're a smoker, maybe not much of an issue for people at BYU. Gender, more women than men have the disease, although there still are a lot of men that get this disease, and but just slightly elevated risk if you're a female. Sunlight exposure is another player, and I'm going to talk to you specifically about this and some hypotheses about why sunlight exposure may be problematic in this disease. And then, just like sunburns, if you have more pigment in your skin, you're more protected from burning. The same is true in your eye. If you have blue eyes, you're more at risk for the disease than if you have brown eyes. So this woman right here is doomed. What she really needed to do was at least put some sunglasses on and take the cigar out of her mouth, and then maybe she'd be okay. But, she's pretty much got all the risk factors that we have here. Okay, so there are two main types of AMD. So if you're diagnosed with the disease, you may be told that you have one form or another. The wet form, or the neovascular form, is the more severe advanced case. And I'm going to go into more detail about this in a moment. But the other type of AMD, which represents actually most of the cases of AMD, is this non-neovascular dry form. So the wet form represents about 10% of the cases, and as I mentioned, it's the advanced form of the disease. So 90% of the blindness associated with this disease is seen in this group of 10% of the people who have wet AMD. And what happens, and I'm going to show you a picture of this next, is that blood vessels begin to build up under the eye, and it pushes the retina up so that a lot of scarring begins to form. And as a result of these blood vessels forming, they'll leak, and rapid damage occurs to the macula. So this is a photo, and as you see here, those are the abnormal blood vessels. That is the retina, which is about 7 different layers of cells, and it's literally pushed up by the blood vessels that are leaking there. Here's another picture. Unfortunately, it's hard to make out on this screen because retinas are red, but you can see this is a normal macula. That's the optic nerve right there. That's the macula. This is the whole retina back here. And you can actually make out the blood that is building up as a result of these blood vessels breaking. And so when you go to the ophthalmologist and you have wet AMD, they're able to take these photos and diagnose it quite quickly because they can see with this what is called fundus photography and diagnose people this way. Okay, so on to dry AMD, which is actually going to be the majority of the talk, because what I'm focusing on is research related more to dry AMD, which is the early stages of the disease. So 90% of the cases are dry, but only 10% of these result in blindness. This is the early stages, as I mentioned, and our ultimate research goal is to stop AMD before its onset or within that dry stage. Because you may be aware there are some treatments for wet AMD. There's a drug on the market. The problem is once you've advanced that far, because the cells are non-replicating, you might be able to stop it at that point, but those damaged cells aren't going to come back. So if we can stop the disease earlier as a result of understanding the mechanisms a little bit better, that would be the best case scenario. Okay, so here we've got some retinas, and imagine that these are red. And again, this is your healthy, normal macula. The macula is in the center there. What ophthalmologists are able to detect when somebody has dry AMD is these yellow patches in the eye. And I'm going to talk to you about the chemical composition of these yellow patches, because that's what we are studying in our lab. And the yellow, the fluorescence that you see here, is a result of something called lipofuscin that builds up in the eye. And we're going to go over this here in a moment. So, on to the visual cycle. I just want to mention this briefly, just to let you know, why are we able to see? What happens in the visual cycle? What happens is there's a vitamin A shuttle. So this is vitamin A right here, the structure of vitamin A, a carbon-based molecule. And it gets shuttled between two of the cell types in the retina. So as I mentioned, there are 7 layers of cells, and the outermost layers are the photoreceptors and the retinal pigment epithelial cells. And what happens is vitamin A gets shuttled from the photoreceptors to the retinal pigment epithelial cells. Then it goes through a number of chemical changes, goes back into the photoreceptors, interacts with a particular protein in the eye, and this leads to a signaling cascade that results in a communication with your brain that leads to vision. So this is what happens when we have good vision. This is the healthy process. Okay, so these are the cell types in which this visual cycle occurs. You saw those pictures earlier of the yellow spots that accumulate in damaged retina, and that is represented right here as these orange spots in the retinal pigment epithelial cells. These are lipofuscin granules, fluorescent granules that build up in the eye. And as you see, again, this is in the retina, the back part of the eye, and what happens, the reason why this lipofuscin builds up over time has to do with some of these chemical processes involved in this visual cycle that I showed you. So if we look here, first of all, those cells, those are the retinal pigment epithelial cells. They're non-replicating cells. Your photoreceptor cells are like fingernails. And the retinal pigment epithelial cells, we use the photoreceptors as nutrients, and they essentially engulf, as you see here, the photoreceptors and enzymatically degrade the materials contained within the photoreceptors, use them for nutrients, and then anything remaining in the photoreceptors for which there is no enzyme to degrade them ends up getting deposited as these fluorescent yellow granules that I showed you in the eye. And so this process, we've detected lipofuscin in teenage eyes, teenage healthy eyes. So the point is, this process begins in our youth. And the reason why the disease is associated with age is because eventually enough of this material, which has nowhere to go, essentially cellular garbage, just accumulates in the eye and it causes damage. Okay, so here's another photograph of lipofuscin, and this is actually a real photograph of lipofuscin as opposed to the diagrams that I showed you earlier. And this compound right here is one of these vitamin A derivatives that I talked about at the very beginning, and the type of compound that we work with all the time in our laboratory. So this specific compound is made up of 2 molecules of vitamin A aldehyde, of an oxidized form of vitamin A, and phosphatidylethynolamine. So what is phosphatidylethynolamine? Well, it's something that makes up a cell membrane, and because of problems that occur in the visual cycle, such as too much vitamin A being cycled through, some of that vitamin A, instead of being cycled back into the cycle, will end up reacting with this phosphatidylethynolamine to form this compound. So this compound on its own is not particularly toxic, but it's when this compound reacts with light that there are problems. So that's what I was telling you about at the beginning. We've got these eyes, this most highly oxidized tissue in our body. So we're always exposed to oxygen. And in the presence of light, of course, we're always exposed to light, and light is very important for the visual cycle. You saw that light comes in and it actually, is part of that important process of vision. But too much light and too much vitamin A lead to problems resulting in cell death. And that's what I'm going to continue to talk to you about. So, A2E versus lipofuscin. So there were studies before A2E was isolated that suggested that these granules were problematic, especially when they were exposed to light. And so chemists started investigating, what is it in these granules that are causing the problems? And that's when A2E was originally discovered. And lipofuscin has been implicated in the cause of AMD. I know I told you at the beginning the cause is unknown, and so we always kind of tread lightly when we say lipofuscin. We don't say lipofuscin causes AMD, but we say it's implicated in the cause of AMD because research suggests that it plays a role. And A2E, because it is also what we call a phototoxic compound, it reacts with oxygen and light to form toxic oxidation products. It appears to be at least a major player in lipofuscin. However, there have been some studies that have suggested that there are other players that are unknown that may also be in lipofuscin, and that's what we've been trying to discover in our lab. So lipofuscin actually, a lot of people in discussing A2E talk about, lipofuscin mostly contains A2E, but really it only contains about, less than 1% of lipofuscin is made up of A2E. So there are other proteins, lipids, and vitamin A derivatives that are also present in lipofuscin. And a particular study showed that A2E only accounted for 25% of lipofuscin's oxygen consumption. And why is this important? As I mentioned, the cells in your eye are as far as tissues in your body, they're most exposed to oxygen. And lipofuscin, because its mechanism of action or its mechanism of damage involves oxygen and light, if A2E is only consuming about 25%, what's consuming the rest? And are these other compounds or proteins or whatever it may be also major players in the disease? So I mentioned to you this phototoxicity. Now, specifically, if you go outside or, you're exposed to these lights in here, you're exposed to visible wavelengths of light. The specific wavelength of light that is responsible for harmful photooxidation products occurring on this compound is the blue wavelength. So we study these compounds by shining them with blue light and then evaluating what toxic compounds may form. So research has shown that specifically that A2E, and I'll show you some of my compounds as well, form these cell-damaging oxidation products. Also, they specifically damage DNA. So that's one of the problem areas in the cell that leads to the cell death, is this DNA damage caused by oxidation products of these vitamin A derivatives. And ultimately, these oxidation products will result in a programmed cell death. And of course, as I mentioned earlier, if those cells in the retina die, they can't be replenished. Okay, so my research, what we've been doing is we've been looking for new compounds in lipofuscin that may also be involved in AMD. And this library, and what I mean, library is filled with books. Well, my library is filled with molecules. I make these vitamin A derivatives. And I've made this library so that we could specifically use the compounds in this library in order to detect new compounds in the eye that might be involved in AMD. So what we do, there are many different analytical techniques that we use, but I thought this was a visual technique that I could show you a little bit. Uv-Vis spectroscopy. We get this nice spectrum that has these two nice bumps on it, and this tells us a lot about the structure of the compound. And so we're able to actually use this UV-Vis spectroscopy, a type of spectroscopy that utilizes light and looks for changes in the molecules. And we're able to utilize this to detect new compounds that have similar structures. And the reason is because if a compound has Similarly, if it has these two molecules of vitamin A associated with it, it will have these two bumps like this. And so we're able to really use this to help us find new compounds. And let me tell you why we thought that there might be other vitamin A derivatives that are forming in the eye. As I mentioned, this oxidized form of vitamin A reacts with the cell membrane to form the A2E compound that I showed you earlier. There are other compounds like phosphatidylethynolamine, that cell membrane compound, and these are some of the compounds that have similar functional groups that would be able to react chemically with the vitamin A derivative. And some of these compounds you're probably familiar with, like dopamine and serotonin. These are the types of compounds that where we know are in the retina and may also be reacting with the vitamin A. And so what we've done is we've formed compounds that we form these reaction products, and we get human eye tissue and look for the compounds in the tissue. So this is how we do it. We do a synthesis in the lab. We call it a biomimetic synthesis because it is a synthesis that mimics what should actually be happening in the eye, where we react this oxidized form of vitamin A with those different compounds like dopamine and serotonin, and we get a variety of products. Some of them have one molecule of vitamin A associated with them, some two, and some as much as four. And these are the types of products that we've been building in our lab. Okay, so what do we do once we have these compounds? Well, as you saw in the visual cycle, these compounds are very reactive with light. And so we work in the dark in our labs. We work under red light because red light doesn't interact with our compounds. So we work under red light conditions and extract human eye tissue, retinal pigment epithelial tissue, to look for some of these new compounds. So how do we do this? Well, we get the material from the Utah Eye Bank, so from organ donors. And just to give a little plug for organ donation, the nice thing is that we're not the only people that use the eyes, but the corneas are used for corneal transplants. And then another researcher uses the whole retina to do biochemical studies, also studying some of these blindness-related diseases. And then we specifically use just that one layer of retinal pigment epithelial cells to detect new compounds that might be involved in blindness. So these eyes really go a long way. Also, mouse eye cups are used. So there are some what are called mouse models for the disease out there. And we have been involved in, looking at some of these mouse models to determine whether they have the right chemicals in them to therefore be good mouse models for the disease. And we also use these mouse models to help us study the disease, so we have additional tissue to study. And we extract them, as I mentioned, under these red light conditions using organic materials. We call them solvents. And we just sort of grind the material up and extract them and then analyze that material. Now, I'm just going to point out some peaks and tell you why these peaks were interesting to us. The peaks in and of themselves, now what we did was after we collected this material, we separated it. There are a variety of organic separation, purification techniques that we've used. And we separated the material. And the reason why peaks A, B, or A through F were of interest to us is because all of these peaks had that characteristic UV spectrum that I showed you earlier with the two humps. And so we were able to use that and say, okay, these are the peaks of interest here. Let's narrow down to these peaks, collect these peaks, and determine their structures. When you're working with human and mouse eye material, you really have a very small amounts of material to work with. So it's tough to get full characterization on these materials. But we were able to use our standards, which is the reason we synthesize these compounds, and compare them to some of these peaks. And we were able to show that there was indeed a tyramine compound forming. So it was tyramine plus 2 molecules of vitamin A. And this compound it's a dopamine derivative of it's very similar in structure to dopamine. And so just to give you an idea of something you might be familiar with that's forming in the eye. So we were able to narrow down that compound was forming, as well as a cadaverine compound. So using this methodology, we were able to find 2 new compounds that had similar structures to A2E, which may also be major players. And that's where we are with this right now. Now we've done some photooxidation experiments where we shine these compounds with light. We see that they also form these toxic oxidation products. So we have some preliminary evidence that suggests these compounds may be also players in the disease. The next step is to actually look at these compounds specifically in retinal pigment epithelial cells to see if they may be causing similar problems to the A2E compound that I talked to you about. And this is just something that we did with the mouse model eyes. So we were also able to see, and there you see that characteristic UV that I talked to you about, but this unknown compound was also of interest to us because it had that same UV profile. And so as a result of these studies, we were able to say chemically, this is a good mouse model to study the disease because there's more A2E in this compound or in this mouse eye than in just a normal mouse eye. We were also able to show that there were some of these new vitamin A derivatives that I just talked to you about in this mouse model. So the good news about this is the people who designed this model were able to say, okay, because we see more of these chemical markers in the eye that are associated with AMD, and the way they made this mouse model was it's called a transgenic mouse. So they were able to target a particular gene that they thought was involved in the disease. And the reason why people in Utah like to do these studies is because of the fantastic genealogical resources, and families will actually get involved in these studies, and they will be able to work with families who have AMD and look specifically at these gene mutations in these families. And so this is one other approach to the disease. It's sort of a combination of approaches, a genetic approach and a chemical approach kind of coming together. Okay, and the last three AMD slides that I'm going to show you have to do with a study where we worked with a research group in New York, and they had some, well, they specifically do a lot of mitochondrial studies. And so we were able to supply some A2E, some of this compound that is involved in AMD. I hope I'm not overwhelming you with acronyms, but macular degeneration. And in the presence of physiological quantities of A2E, mitochondria begin to dysfunction. So why is this important? Mitochondria are really the powerhouses of the cell. They produce a lot of what is called ATP, which is cellular energy. And specifically what happens in the presence of A2E is that this ATP doesn't form as readily. And so this is one of the mechanistic studies in which we were involved, and we were able to show that these compounds cause problems in this specific manner. And the reason it's important to understand this is because the better understanding we have of the mechanisms of the disease, the better able we are to design solutions, to design drugs perhaps that may circumvent these problems. And so we were able to see that the mitochondria began to dysfunction in the presence of these vitamin A derivatives. And specifically, when the mitochondria begin to dysfunction, cellular processes also begin to be impaired. One of those cellular processes that I talked to you about earlier, phagocytosis, where the retinal pigment epithelial cells will engulf the photoreceptor cells, those fingernails in the cells, and that specific process starts to become impaired in the presence of A2E. And the good news was that if we treated these cells with antioxidants, we were able to completely inhibit the process. And this means, well, the implications to us would be that we need to eat foods rich in antioxidants. And overall, what happens is the more mitochondrial, we tested various types of mitochondria. Some of the mitochondria was more impaired than others. The more impaired the mitochondria, the more the A2E affected it. So it was this sort of synergism of negativity, I suppose, where in the end there were exponential problems occurring with the cells if the mitochondria was impaired and this A2E was present. But eat foods rich in antioxidants, and hopefully you can stop this process from occurring. So now I'm just going to wrap up the AMD portion of the talk and then talk to you a little bit about this targeted and triggered drug delivery system for cancer therapy. And what have we done in the AMD field? Well, we've synthesized these vitamin A derivatives and isolated new compounds that might be involved in the disease. We still need to do more tests, but we're encouraged by preliminary results that we may have found some new culprits, and therefore we can hopefully target drugs to work against these culprits. And some of those drugs may simply be antioxidants, vitamins that you can take. But nevertheless, now that we know more about the disease, we can hopefully target it better. We also worked with a team to discover this mouse model that is now being used to study actually a juvenile form of macular degeneration. And we discovered these two mechanisms of aging that synergistically work to exponentially decrease cellular function, leading to AMD, and showed that these mechanisms could be completely inhibited. The mechanisms of damage could be completely inhibited in the presence of antioxidants. So what does all of this mean to you? You've probably picked up on some of it. And what can you do to prevent AMD? Eat healthy foods that contain antioxidants. And I just put a list of these foods here, but this is certainly not a complete list. It's just some ideas. Foods such as blueberries and spinach, wheat germ, beans, nuts, asparagus, avocado, artichokes, olives, and even oils contain antioxidants. So try to eat good foods. And of course, specifically if you know that AMD runs in your family, it would be a good idea to take an antioxidant supplement as well. And there are specific supplements that your ophthalmologist or optometrist can actually prescribe you that contain vitamin C, vitamin E, and zinc, as well as some carotenoid compounds that are really good antioxidants and can help fight against cell death as it progresses. They can't build up new cells, but they can at least inhibit the progression of the disease. So make sure that you check your family history. If you don't know if anyone in your family has this, you should check. And because, as I mentioned, that when we've analyzed eyes, we've seen lipofuscin beginning to form in teenage years, it's important that no matter how young you are, that you start eating well, and especially if you know that this disease runs in your family, it certainly wouldn't hurt to take a supplement. And then lastly, wear sunglasses. I wanted to mention, even beyond AMD, that it's good to wear sunglasses if you're exposed to blue light. These seasonal affective disorder lights that some people use are blue, and sometimes you just expose your eyes to them. In dermatology, there are dermatological devices that they use blue light. And so really just be careful around blue light, or all visible light, but specifically these blue light apparatuses. And if you're playing outside in the sun, it's good to wear sunglasses. Okay, so this part of my talk is much briefer because you already understand a bit about the way these compounds work, the interesting activity associated with these vitamin A derivatives, that they're relatively non-toxic in their unoxidized form, and when they're exposed to oxygen and light, they form what we call cytotoxic oxidation products. They form compounds that damage the cells. And as I worked with these compounds, I started thinking that we could utilize these mechanisms of damage to build a drug for cancer. And the idea behind the drugs that we're developing is that we attach this compound to folic acid. And the reason we do this, and I'll show you a little diagram here in a minute, but cancer cells, certain types of cancer cells, actually have more receptors for folic acid than regular cells. And what this means is you can target compounds with folic acid attached to them, specifically to the cancer cells. And then, of course, we've got another part of the drug, which is the trigger. And the trigger for the cytotoxic oxidation products is blue light. Okay, so we performed some photooxidation experiments, and these experiments were useful to us both for the macular degeneration part of our project in order to determine if these new compounds that we had developed actually might be also causing problems in the eye. But we also did these photooxidation experiments to look at different reactivities of the compounds that we built in our library, and we were able to see some compounds form more of these oxidation products than others. And all of the compounds that we've tested have had these 2 molecules of vitamin A and then some type of amine like dopamine or serotonin, like I talked to you about earlier. And so we exposed these compounds to blue light, took samples, and analyzed them to look at the oxidation products. So this is called, we used mass spectrometry to analyze as well as other techniques. But this is the molecular weight of our parent compound. So here we see, okay, we only have the parent compound. Then we shine this compound with light, and after shining it with light, you see all of these peaks here? These are 13 potentially cellular damaging oxidation products that are forming. And some of these compounds are forming, these particular compounds that I show you right here are actually, they're cutting the compound in half and adding oxygen to it, and adding oxygen to various bonds in the molecules, and it's this oxygen that when it's attached to these compounds that causes problems. So we're able to analyze and see, okay, that compound forms 13 potentially cellular damaging oxidation products. And just to compare it with one of our other compounds, we looked at this compound right here. Doesn't have a whole lot of difference structurally other than an extra oxygen and carbon, really, and a couple of hydrogens. But we saw dramatic differences when we shined the compound with blue light. We saw after an hour only 3 oxidation products formed. Remember, the other one had 13 products forming after an hour. And then after 4 hours, only 4, and 5 hours, 5. And after that, we weren't really able to see much. So we then did some experiments in cells. So that information we're able to use to say, okay, these oxidations are forming in this way, and these compounds react differently. But it's really when you do experiments in cells that you're able to determine which compound might actually be a better compound as an anti-cancer agent, or which compound might be causing more damage to the eye in macular degeneration. So we used some leukemia cells, and we incubated this compound. What that means is we just put the compound into the cells and we heated it up to your body temperature. And then we shined the cells with light and we looked at what happened to the cells. So we were able to see this is with that compound that had the 13 potentially damaging products that we saw by mass spectrometry. Here we were able to use just 2 different assays. One, we showed 71% growth inhibition. So here, this is where we don't shine the cells with light. We don't see anything happening. So the compound that is unoxidized doesn't cause damage to the cells. But when we shine it with light, 71% of the cells die. You can think of it that way. And in this case, 83% of the cells die. So we were able to show that this compound showed 13 damaging oxidation products. We put it in the cells, and it also caused problems. And these were cancer cells, so we wanted to kill these cells, unlike the cells in the eye. Okay, so A2EE, this is the compound that had only 13 damaging oxidation products, only 3 damaging oxidation products after 1 hour. And here we saw, after 30 minutes, only 20% growth inhibition, so 20% cell death. So okay, it works a little bit, and we saw some oxidation products, but we were able to show that if we could look on mass spec and see that a compound had a lot of these cytotoxic oxidation products forming, that in the cells we would see comparable damage to the number of oxidation products. After 50 minutes, we saw more cell death, about 50%. But we really had to pound on this with light in order to get the growth to be inhibited of these cells. Okay, preliminary in vivo, very preliminary in vivo data. We treated a mouse tumor with one of our vitamin A derivatives, and we shined it with light, and after a couple of days we saw that the tumor had decreased in size. And so that gives us even better information that shows that, because it's one thing if you are able to do this in a test tube, it's another if you're able to show some damage to actual cancer cells. But when you can actually put it in an animal and see some damage, then okay, this is promising. And as I mentioned, those assays that we did were all on the compound that wasn't attached to folic acid. But we then made compounds that were attached to folic acid to see if the reactivity would be similar. And as I mentioned, healthy cells only have, a few receptors for folic acid compared to cancer cells. So some cancer cells have as much as 1,000 times the folic acid receptors as healthy cells. And so the idea is that we react our vitamin A derivative with folic acid, we form what is called a bioconjugate, treat it then expose it to the cancer cells, incubate it with the cancer cells, and we should find that more of this builds up in the cancer cells than the healthy cells. And the point we are now, we have made these compounds, and we're about to test them on the cancer cells. So we were a little concerned that folic acid would be too reactive with the oxygen, or that the folic acid would actually act as an antioxidant, so that our compound wouldn't form these cytotoxic oxidation products. But we were able to show, and here I can just visually show you, these compounds that we start with have this rich orange color. And you can tell visually that they've reacted with light just by color change. So we were able to observe that here. And so overall, we were able to show that this photooxidation of these folate derivatives would actually occur just by ambient light. So just light in a room would cause it to occur. So we didn't even have to use our more powerful blue light source. So they actually turned out to be even more reactive than our aminoretinoid compounds or vitamin A derivatives that weren't attached to folic acid. And so what that means ultimately, and we're still working on this, is that less drugs should be needed, and that would mean less toxicity for the body. And the idea of targeting the cells is so that we kill the cancer cells and not the healthy surrounding cells. So that's why we're trying to develop this type of drug. Because a lot of chemotherapy methods now, as you see, people lose their hair and nails, and that's because the drugs not only target cancer cells, but other healthy cells as well. So overall, we built these vitamin A derivatives with and without folic acid, and we studied human and mouse tissues and developed a mouse model for the disease. We also discovered 2 new mechanisms of aging that really cause damage to the macula that can be reversed in the presence of antioxidants. And we looked at these vitamin A derivatives in solution, in vitro, and in vivo. So in as you saw, the solution, the test tube, the cancer cells, and the animal, and we were able to see activity in all of these. So I also, just before I end, I have to acknowledge I'm not the person in the lab doing all of this work. I've had lots of help. I have collaborators, Paul Bernstein, Sylvia Finneman, and Kang Zhang, whose work I've talked about today. They're collaborators on these projects, as well as specifically Junping, Mackenzie, and Mary. They did a lot of the work that I talked about today. So, and I'm also grateful for the funding that I've had for these projects. And of course, grateful for all of you for showing up today. So thank you very much.